Thank you very much, and thanks to the organisers for inviting us back. It's been a busy year at Race. I'm going to be talking about some of the developments there, and hopefully it's going to be a very exciting year ahead. And just, after Michael's talk, Arabella, I lost a next-door neighbour yesterday to gastric cancer, and it really sort of underpins in my life why we get up every day and do what we do with the support of people like yourselves. And we've got a few shareholders in the audience, so good to see them, and hopefully we'll get a few more today. So, right, Race Oncology, at the heart of cancer care. As I said, it's been an exciting time for the company. Very quick snapshot, ticker RAC, market cap's around 200 million. We've got just under 14 million, or had just under 14 million in the bank at the end of the last financial year. Only burnt 4.5 million. We really, controlled costs during that period of time. So we've got enough money in the bank to see us through to the end of 2027 and to fund all of the programs that I'm going to be talking about today. So why invest in Race Oncology now? I'll just give you a brief snapshot. I'm going to fill in a lot of the details along the way. The number one thing I want to emphasize is that the active pharmaceutical ingredient, the API in our drug, RC220, has been extensively tested back in the 70s and 80s. And so, there was talk earlier on about what you need to do to get your drug onto the market. We actually know this drug works because it has worked repeatedly in the past across a very broad range of cancer indications. It was never launched because it had a problem, and what Race has done is solved that problem and generated a new drug, RC220, which also has the benefit of having fresh intellectual property. So we've got a 20-year patent life now running on our drug, RC220. We've also shown that whilst it's a potent anti-cancer drug, it can actually offset some of the toxicities caused by other cancer drugs. And this is actually an increasing focus out there in the oncology world, trying to make sure that patients, well, obviously survive their cancer, that's number one, but number two, and now an increasingly close number two, is to make sure that those patients are left well, functionally able to continue and have very long, happy, healthy lives. In terms of Race, we've just started our clinical study of RC220, old drug, new formulation, and we should be generating a lot of data through the next 6 to 12 months. So a lot of value drivers coming from that. And there are some things we've been working on in the background that we hope to be announcing along the way, which I cannot talk about today. So let's get on to it. So as I said, we're really focused on trying to improve the outcome for cancer patients. More cures, longer remissions, but also leaving patients healthy at the end of their treatment. And a lot of them are not, unfortunately. So let's just take a stop here and talk about the original drug, bisantrene. As I said, it was developed by a company called Lederle, which now forms part of the Pfizer group. It was developed in the 70s and 80s, and it was actually approved for acute myeloid leukemia in 1988 in France. It had showed excellent outcomes across a very broad range of cancers, both liquid cancers like leukemias and solid tumours as well, like breast cancer, ovarian cancer. In fact, in a number of phase 3 studies, it bested the standard of care, so drugs like doxorubicin, which is very widely used. But it had one flaw, and that was it was very insoluble, and it was very, very difficult to administer to patients. If you actually injected it into a peripheral vein, as soon as it got to physiological pH, it would crystallize, causing immense pain, phlebitis, and there are stories from that era of patients on trials ripping catheters out of their arms, because of the pain. So it had to be delivered by a central line, which basically is a catheter that goes into veins returning blood to the heart, because then it is immediately into a large volume of blood. And that's how they developed the drug for AML. So right at the end of this extensive clinical testing period, they decided they weren't going to commercialize it, and it was dropped. So what we've done at Race, using, Lederle clearly would try to have made the drug soluble. They tried for many, many years to do that. We've spoken to some of the people who actually worked on that program. We have, at Race, have applied modern technologies for solubilizing drugs, and we've created RC220. It wasn't very easy, but we've managed to do it. It preserves the same profile in the blood, so pharmacokinetics, pharmacodynamics, that's how much drugs in the blood and where does it go. We basically recapitulated the old formulation, but we can deliver it by a peripheral vein, and that's the main difference. I mentioned the new intellectual property, and the other thing we're doing is we're applying modern technologies, modern omics, genomics and other omics to look at how this drug works. And we're starting to find some very, very interesting things. Again, we'll be talking about that more in the coming months. But most importantly, RC220 builds on all of that historic data that was generated and ultimately resulted in approval for acute myeloid leukemia. So, I think it should be reasonably obvious by now why we need to protect patients from their cancer treatment. And it's not just chemotherapy, radiotherapy, surgery, all of these treatments leave their mark. Some of the new immunotherapies are more benign in that regard, but particularly childhood or sufferers from childhood cancer, they are left very seriously impaired in terms of them being frail, having a lot more chronic medical conditions as they age. Cognitive development is impaired as well. So these treatments are nasty, and we need to do better. So I'm going to stop and talk about a very, very widely used chemotherapeutic. Again, it's a very old one. It was, I think, first launched in the early 70s. It's a drug called doxorubicin. It is still very widely used today for one very, very simple reason, and that is that it's very, very good at killing cancer cells. So it works across a very, very broad range of cancer types. But it was recognized that it had a problem, and that problem is that it damages the cardiovascular system. Initially, we focused on the heart, but now we know it's actually the whole cardiovascular system, and in fact, probably the whole body is being adversely affected by this drug. But we notice it in the cardiovascular system because a lot of patients, particularly as they're older, are actually using their cardiovascular system at the maximum capacity, walking upstairs, going to the shops. And there was a study done, it was the Peter Mac in conjunction with the Baker Heart Institute, and they use a measure called VO2 peak, which is measuring oxygen, carbon dioxide exchange whilst the patient is exercising, not VO2 max, which is where you run as fast as you possibly can. These are cancer patients, and a lot of them are older, but it's a very, very accurate way of assessing the cardiovascular performance of the patient. And even after a single dose of doxorubicin, they could measure a reduction in the overall cardiovascular fitness of the patients. And it doubled the number of patients in the study who had functional impairments, i.e. they couldn't walk up the flight of stairs or they couldn't walk down to the shops. And so the decline after just 4 cycles of doxorubicin, which is the maximum that you're going to get because of the fear of you having heart failure, patients were losing around 10% of their overall cardiovascular fitness, which is very, very significant, particularly in that older patient group. And that is equivalent to a decade of aging. And we believe, based on the work that we've done, that actually it is aging all the cells in the body, all the non-dividing cells in the body. The drug's killing cells that are dividing, like cancer cells and cells in the hair follicles, but it is also damaging non-dividing cells like those in the heart. And if you damage the heart, that's it. It's damaged for life. There's no coming back. It can't repair itself. And this is what it looks like in animal studies. So on the left-hand side, you can see the heart. This is in a mouse, of course. We can't do this in humans. The heart tissue looking beautifully pink, as it should. In the middle is the heart from mice treated with doxorubicin, and you can see those really marked grey flecks. That's collagen. This is fibrosis. We're going to hear more from others talking about fibrosis later on today. But that is scar tissue, basically, in the heart. And I think it's fairly obvious that that is not going to be helping your heart to pump more blood. It's going to be doing quite the converse. On the right-hand side is our drug, 220, in combination with doxorubicin, and just visibly it looks as if there's significantly less damage. And this is what we see across a range of different models, and we've tested the drugs in combination on human cardiomyocytes, and we see the same effect. This is what we're trying to prove in our upcoming clinical trial. Now, importantly, if we put our drug together with doxorubicin, we definitely do not want to blunt that anti-cancer effect. As I said, the number one thing that patients want, the number one thing that oncologists want, is to get rid of that tumour or to keep it in remission for as long as possible. When we put RC220 in combination with doxorubicin, across 85% of the cell lines tested and 85% of the cancer types tested, we saw an increase in activity. So doxorubicin is very active, but putting our drug 220 with it increases the cancer kill. The 15% of the cell lines where we didn't see an increase in activity was because the doxorubicin was already so active, it was very difficult to see a marginal improvement. So I think it's really important that we're combining that protective effect with an increased anti-cancer effect. So what's the market opportunity of this? We get this a lot, that people say, well, chemotherapy, it's an old treatment, nobody's using it anymore. Actually, the use is increasing. And quite a stunning number is the little circle on the left-hand side. That's sales of doxorubicin. Now, doxorubicin's been off patent for decades now. It's manufactured by a lot of generic companies who compete on price, and so it's about $100 a vial. But still, even at that very, very low price point, $1.5 billion of doxorubicin used every year. Now, if we manage to get RC220 approved for use in combination with doxorubicin, it's going to get a new drug price, something around $15,000 to $20,000 US, according to research that we commissioned. And to get to that figure from the Triangle research report that we commissioned, to get to $7 billion, that represents 3% of doxorubicin use globally being converted to use in the combination. So hopefully that's not too heavy a lift for us. So on to the clinical programs. We recently published some data from a study that we supported in Israel. There were 2 studies done, one as a monotherapy and one in a combination therapy. And I mention this because this is new data from the modern era, proving that the data that was obtained historically by Lederle is still relevant today. I'm not going to say any more about that. We are discussing the potential to continue to advance RC220 in AML because of its very high level of activity. But the trial, RC220, the cardioprotection, which is the third one down, this is the trial that we've just started, and the first patient has been dosed, and I don't think we said publicly any more about that, but it's going well. This is a 2-part study. First part, on the top row, part 1, is a dose escalation. So the patients will get a dose of RC220, and then they'll get a dose of RC220 in combination with a full-strength therapeutic dose of doxorubicin. So the patients are definitely going to be getting something therapeutic. And then we go 3 patients at the lower dose of bisantrene, and then the next 3 patients will get a higher dose of RC220 in combination with doxorubicin. Once we've identified the maximum tolerated combination dose, we will then expand the cohort and start looking at measures like VO2 peak, measuring the health of the patients, looking at markers of aging, looking at kidney function, etc. So a whole range of different measures and markers to establish how our drug is working in combination with doxorubicin. So in terms of upcoming milestones, I think we need to update this slide. We've actually achieved quite a lot. A lot of these milestones were to do with the setup and initiation and the start of recruitment of patients into the study. We've just got 2 sites open so far in Australia. We've got ethics approval for 2 sites in Hong Kong, and we have 4 sites that will be opening in South Korea. We actually expect, because of the way that doxorubicin is used, that we will actually get most of the patients from Hong Kong and South Korea. So once we get those sites up and running, that's when I think we'll see a real uptick in recruitment into this trial. So I've got a few seconds left. So the key highlights I want to leave you with is that Race is developing a substantially de-risked asset in that the active agent in RC220 is very, very well known, it's known to work, and it's known to be safe. And so that reduces the risk of failure. It doesn't eliminate it completely, it's got to work as we predict it will in combination, but we know it's active, we know it's safe. It's solving a real problem, and that is there are still patients who aren't treated optimally with drugs like doxorubicin, or patients who don't receive doxorubicin because they're judged as being at cardiovascular risk. So we think we can expand the use there or increase the number of cycles that can be given. We're not going to be creating a new market. We're going to be converting an existing market. That's for doxorubicin. and we're going to be looking at expedited pathways, and I've got 1 second left. So I'll stop there. Thank you very much.